What's going on, GeneGuard family, and welcome to another episode of the Biotech Startup Series. Today, I'm speaking with Jacob Becraft, the co-founder and CEO of Strand Therapeutics. We're going to be discussing mRNA therapies, biohacking, and many, many more. Stay tuned. So, Jake, thanks for coming to my show. It's really great to have you here.
I'm happy to be here, man. Thanks for having me.
So, before we go into Strand Therapeutics, please tell us a little bit about your background and your professional experience.
Yeah, so I started my career in science as a PhD student with Ron Weiss at MIT. So Ron is the director of the Synthetic Biology Center. He focuses on genetic gene regulation technology, right? So synthetic biology as a way to control how genes are expressed, make, I guess Ron's overarching goal is to be able to program cells as if they were computers. He comes from the computer science realm. He's one of the first synthetic biologists back with Tom Knight, who co-founded Ginkgo. Back in the 90s, and then his new field is can we bring this sort of programmable genes into the new field of gene and cell therapy. And I went into his lab and wanted to build better gene therapies. I wanted to build therapeutics that could be encoded with any sort of a therapy for a patient, but I wanted to build them so they were smarter, so that they could go into a patient and kind of, if you have a tumor in your body, you could inject this into the patient, and it could, the gene would go all over the body, but it would never be active. It would never turn on. It would just be pretty transient and eventually dissipate unless it found like a tumor. And then if it got into that tumor, it could activate and attack and cause, whatever that therapeutic sort of outcome is.
Okay, got it. So basically these genes that you're trying to, target tumor cell or other types of cells of interest, are these like completely synthetic, or are they known proteins or novel proteins? What's your approach?
It's really everything under the sun is possible, right? So you can use what's called endogenous proteins, like actual protein that is in your body, and then we just encode a synthetic version onto the messenger RNA, and then deliver that into the body in order to, either fix an issue with a gene that is mutated or not functioning correctly, or to sort of, kickstart a response in a certain way. Or you can take a different approach, and you can encode like a synthetic antibody, for instance,
right.
And then have that delivered into the body. Or you can, like in the case of COVID, you can encode a piece of the actual COVID genome, the spike protein, and use that to train the immune system and actually create a vaccine against COVID, or against any virus, really.
Okay, we're all pretty familiar now after over a year now of this debacle with COVID. But, obviously the COVID and the COVID vaccines made mRNA in general extremely popular. So it's a big hype now. But Strand Therapeutics was founded way before coronavirus, way before, any kind of approved vaccines or therapeutics with mRNA. How did you envisioned that this is going to be the future of therapeutics or vaccines? Or why did you take this route? Why not, let's say, microRNAs or the interference pathways? There are other nucleic acid types of approaches. What drove you more to the mRNA?
Yeah, of course. So my thought has always been that fundamentally what we're trying to do with any sort of gene therapy, right? What we're trying to do is take a gene that we design, whether it's one that we copied from the human genome or one that we've designed in a lab that creates a protein that has some sort of functional effect in the body that can mitigate some sort of disease. And we're trying to get that gene into a certain area of the body, and in order to either correct a default or kind of enhance an underlying feature, create an anti-tumor environment, whatever that is. And so when I came to MIT in the fall of 2013, I met the man I eventually co-founded Strand with, which was a scientist, a postdoc in Ron's lab by the name of Tasuku Kitada. And Tasuku I came to him and said, I think the future of medicine is going to be in this encodable gene kind of avenue, either, engineer viruses, engineer synthetic DNA. But I think that, the information nature of it is such that it's such a leap forward, right? At the time, all we had was the ability to make synthetic proteins in the lab, try to inject them into people's bodies in order to solve some sort of disease issue. With this genetic technology, it was very much like software, right? So you built a system, an operating system, and then you would drop in different sequences of different proteins into a parent system, and that would allow you to create, just an incredible amount of therapeutics, all off of kind of the same manufacturing line and the same process. And so that caught my attention. But when I came to MIT with that idea in my head, Tasuku actually was pretty instrumental in kind of telling me about this new field of messenger RNA. And we hit it off, really got along well together too, I think, which is important, right? When you're running a startup company, it's important to have this sort of like working relationship. And that was a big part of it too, right? We clicked, we saw the future similarly, and we worked well together. And that kind of dragged me. I would say Tasuku really more than anyone is, more than myself, is responsible for, any of the early successes of like my scientific career. Really carried me along in graduate school towards this area of messenger RNA. And the more I worked on it, the more I became convinced that this was such a revolutionary way to develop medicines that it would be the future. But it needed new technologies, right? The technology that Moderna and BioNTech and the people who have developed the COVID vaccines on mRNA were able to develop was fantastic. But it was really fundamentally limited. It was kind of going to go up to, vaccines. And I felt like right at the vaccine level was about as far as they could take what they had without further advancements. And so we started building, at the time, kind of in my head in 2013 and 2014, I was like, it'd be so cool if we built something of value. And then, Moderna or BioNTech contacted us because we knew those companies pretty well. The field was pretty small at the time. Mm-hmm. And, if Ugur Sahin, the CEO of BioNTech, gave me a call and was like, hey, I want to in-license your technology. And I want to build your technology into BioNTech's platform. But the way that things kind of shook out, it made more sense. The more I understood about the biotech field, the more experience I got, interning with venture capitalists and learning about startups, the more it made sense to me that the company actually needed to stand on its own. It's so differentiated from even the other mRNA platforms. So we spun the company out, and really I've been a proponent of mRNA ever since. I think the pandemic has really done is it put us in such a situation where mRNA vaccines were really poised to show off what everyone had been kind of promising.
Right. So I have two questions. So you mentioned that you're totally different from Moderna in the vaccine field, which is, I kind of, know why. I'm thinking delivery system, first of all, and target specificity. I think these are like major issues in general. And the way you're trying to tackle it is not like, the delivery system doesn't have to be specific, but the payload, it should only detonate where you want it to detonate, not everywhere it gets. I think, that's pretty clear. But I'd like to understand one thing about this mRNA platform that you're developing. Is this like one therapeutic equals one mRNA, so one gene product, or is it like multiple? So when you're like dosing a patient in clinical trials, when you get there, is it going to be like multiple mRNAs combined to create that therapeutic, or just one? How does this programming work? It's just one piece of code that can like restructure everything in that cell?
That's a good question. So the first products that we've made that are going to be entering clinical trials in the near future, those are, just a single messenger RNA. Now there's a lot kind of tied into that messenger RNA. There's a lot of different programming, sequence optimization, how those things work. It's very advanced. But I think as we go beyond that, right, as we jump into new and more advanced types of messenger RNA, some of the stuff we're working on in-house inside the company has all sorts of different functionalities, and all, I would say nothing is really off the table with what we can do with messenger RNA. I think the sky is the limit, and our imagination is the limit. And very luckily, the field of messenger RNA has been a renewed interest among investors as well. So that's been really exciting to see as well.
Great. Yeah. That makes sense. Now, in terms of developing these molecules, what's your process? I mean, are you using some sort of computational platform? And how are you, getting to the sequence, to the protein that you're trying to? Are you going from the target all the way to the mRNA, or you have a different system?
Yeah. So, I mean, there's a lot of different pieces that go into designing a drug, right? There's, especially when it comes to messenger RNA. So there is, what's called the backbone of the messenger RNA. So everything that is kind of not the, so maybe it makes more sense to start with the protein itself. So the messenger RNA is delivering a sequence of a given protein, right? A given protein that we want the messenger RNA to go into the cell and express. And so that is its own thing. So you design that protein, what sort of domains you want it to have, and then we have a number of computational tools that help us actually reduce that protein into a sequence of nucleotides that can be made out of messenger RNA that code for that protein. And then outside of that coding region, there's also what we call the backbone. And so the backbone is all the sequence of messenger RNA that is part of this long strand of messenger RNA, no pun intended, but that can, that's kind of the business end. That's what determines all the different programming and, the cell type specificity and how the RNA is processed and moves throughout the body. And so all of that sequence also has to be designed with kind of, where do you want these mRNAs to be expressed? And so all of that is boiled down into a single sequence of nucleotides that contains, all of the programming. You can think of it almost like compiling, right? When you design software, you're working on a bunch of different modules of the software, and then it gets compiled down eventually, and that is into like the final sequence, which is, some that a computer can process. All of that we compile down into it's a quaternary or whatever. It's a 4-part code made up of the A, U, G, and C, and that then is synthesized into messenger RNA and then formulated into a nanoparticle and then eventually delivered into a patient.
Okay, got it. And what's your current status of development? Are you testing some lead candidates on cell lines, or what's your current?
Yeah, we have a drug. We've a lead drug that's been actually partnered with Beijing, a company of larger biotech pharmaceutical company focused on oncology that mainly operates in China. And so we had a partnership with them on our lead drug announced recently, where Strand will be developing the drug in the United States and in Europe and in Japan, and Beijing will be developing the drug in China and in Australia and in New Zealand and a few other countries that they're interested in. And that's a great partnership. They're a fantastic partner, and that helps us as a very small company really have a much more resources available to us through the collaboration with Beijing. But further than that, I think we have other sorts of drugs that we've developed. Ones that I think will revolutionize areas of like cell therapy. Right? When I think about cell therapy, CAR T, for instance, chimeric antigen receptor T cell therapy. You take the T cells out of the body, you ablate all the T cells in the patient's body that are left, all the immune system. You take the T cells into a lab. You add some virus that puts a synthetic gene into the T cells, you expand them, you put them back into the patient, and then you hope that those T cells are now armed and can attack, a cancer or whatever it is that you've built a chimeric antigen receptor against. But that's a very complicated process. It's incredibly expensive, and it's very difficult to sort of scale that innovation. And when you think about it at its core, right, from I think what Elon Musk calls first principles engineering, right? You think about what is the core of what you're doing. All you're doing is putting a gene into a cell, right? You're putting a gene into a T cell. You're doing all of this rigmarole because you can't do that very specifically inside of the patient. Now with new technologies, though, perhaps we can, right? Perhaps we can get into the T cells in the body and create an environment where those T cells are expressing our sort of live cargo, right? And so if we take the same chimeric antigen receptor, we put it onto a messenger RNA, we put that messenger RNA into the body and we program it so that it only expresses inside of a T cell. Now we're in a different paradigm, right? We're in a paradigm where that cell therapy that costs hundreds of thousands of dollars and weeks to manufacture and is incredibly unscalable. We're creating an area where now that has just become an off-the-shelf messenger RNA drug.
Yep. Yeah, that makes perfect sense. And with the production of biologicals that's just booming now, like Twist Bio, for example, I know you can just order, like from Amazon, you can just order your sequence upload. It's unbelievable. But that leads me to another topic, right? If you can do that, if technology gets to the point where, basically anybody from a garage can just order RNAs or DNAs and just select the vectors like viral vector or custom vector or whatever vector. What are the long-term issues that you see with this type of liberalization of biotechnology? Yeah,
I think that it's something that people are already actually very much ahead of, right? I mean, you can't just call up IDT, the number one supplier of DNA or Twist for that matter, right? You can't just call them up and order virus sequences, right? Because the moment that those sequences enter, so all of the suppliers, for instance, of synthesized DNA at the time, have these algorithms that identify different sorts of viral elements, and they flag your sequence if it looks like you're ordering something from that has some of these viral components in them, and there are all sorts of levels of security in order to get those. I mean, for instance, like some of the elements that we use to design our messenger RNAs are from certain viruses. And so, all the time, every once in a while, we will get we will get pinged. We go into IDT or Twist or someone, we order the DNA, and it sends us a note and it says like, hey, like you just ordered a piece of a virus, you need to complete the following security steps in order to prove that you're not some terrorist organization, or you're not just some guy in a garage, right? We have to get actual level approval, to say no, this is a biotech company that is run by, some of the leading minds in synthetic biology. This is our research plan, and then you get approval and you can move forward with that. I think but it's something that we need to stay on top of. I think that it's all the same risk that happened when there was a democratization of technology, right? When silicon wafer technology and chips and chip construction went from being, only accessible to the superpower militaries of the world,
right,
or 20th century, into kind of personal computing clubs, people starting to build their own computers, then the internet and how information technology, it's something that every time we add new technology, the democratization, the gains of the democratization and the innovation that happens when more minds are kind of put to a task and able to do all these sorts of things, and we lower the cost and the barriers of entry are high or are lowered. The innovation is high, but we have to continue to think about security, right? With quantum computing, if quantum computing becomes a thing, it's possible that all of our existing encryption technology will immediately become useless, right? And so,
right.
does that mean we shouldn't build quantum computers? Or absolutely not, right? But it means that we'll have to be thinking about quantum encryption, right? And so the same thing with democratization of biotechnology. I think there are irresponsible people out there. I think some of the, the DIY biohacker sort of pseudo counterculture folks. I think it's, the more I see about that, like the series on Netflix, for instance, the more I see people who like to, who want to be a messiah themselves rather than doing anything that's actually scientifically legitimate. But that'll always be a problem.
Indeed, and I know exactly what you're talking about. I know this guy that's experimenting with CRISPR and gene editing in his house, and he's like injecting contaminated substances, and he's like editing genes so like that he gets more muscle mass and stuff like that, and he obviously got severe infections and septic shock and all sorts of really
that's
bad
what he
stuff.
says, right? But in reality, what he's doing is basically nothing, right? I mean, you can inject CRISPR into your muscle and like you're probably just going to get an infection,
Right.
a bit of swelling, like it's not going to change all your muscles anyway. It's going to change this. What are you going to have, like a tiny little bit of extra bicep, like right here? So I think that those guys, when I see people like that, it makes me think more that these people want attention. I think it can be dangerous. I'm all for democratization, opening doors. I think much like we've done with makerspaces, with things like 3D printers and other sorts of technologies, we've created community centers where people can come and learn about them. I think we should do that with biology. But I also think that this idea that that someone should be injecting CRISPR in their garage, when, I mean, truthfully, I've listened to, I think we're talking about the same exact person. I've listened to him speak before. He doesn't know what he's talking about. And what honestly concerns me about it is that because of the way that research works in this country, because of the way that the FDA works in this country, and because of the history that gene therapy has already had, right? Missteps that go into a certain direction cause a clamp down in regulations, and a clamp down, which then has a domino effect onto investment, right? Once regulations clamp down, investors pull back. Less investment means less innovation, and if those sorts of things happen, like they happened with Jesse, Jesse, whose last name I'm sadly forgetting. There was a famous gene therapy trial with adenovirus in the late 90s that led to the death of a young boy, of a teenage boy named Jesse. And he and that death led to a huge pullback and a huge clampdown on the industry, which delayed life-saving therapeutics by over a decade. Right, we could have had gene therapies in the early 2000s instead of in the late 2010s, but because of this pullback, we got delayed. Money got delayed and advancements got delayed, and that should have been avoided. Right, it was a messy, unnecessary risk to take with that clinical trial. It wouldn't happen today, and it's a tragedy. And it's a tragedy for that teenage boy himself, and it's a tragedy for the industry and all the people who died in the meantime because therapeutics that could have been available when people were experiencing disease, maybe in 2009, let's say, aren't available until this year, maybe.
Yeah, no, I totally get it. And these people, they attract regulation, and it's a problem. Now, but getting back to Strand, what was the biggest hurdle in starting the company, right? So you came from the university, had a group of extremely talented and scientifically sound people, but getting this sort of biotech technology to the commercial landscape is pretty hard. How did you get there? What did you do?
I mean, we're not there, right? Like we're a preclinical company. We're far from commercial, right? But to launch a company, I think, especially even 5 years ago in the biotech sector, there wasn't as much buy-in. as into kind of younger technical founder CEO types, something that's very common in the technology sector, where you have sort of this, a group of founders who are thought of as kind of like, subject matter experts and very passionate about solving individual problems, and they bring that solution forward in some sort of a manner. I think in biotech, especially in Boston, it had basically become this sort of old boys club, right? Where a certain amount of people were determined that they could be executives, and that was determined by this small group of investors, and that was the end of it. I think in around 2016 and 2017, that started to change a little bit. The perceptions started to change, and where we ended up as a field was, there were more and more investors, mostly from San Francisco, but some in Boston, some in New York, some all over the world, that were willing to back, technical founders, people coming out of academia, ready to start companies. I think more recently, right? I mean, even recently, so very recently, Recursion Pharma, I believe today, IPO'd. Right, and that's a classic story of a PhD student who came out with an idea and built a massive biotech company that is now a public company and very successful. And a lot of investors, mostly San Francisco investors, who were willing to back a young technical PhD to build a biotech company, made an incredible amount of money. The old school sort of old boys club of Boston Biotech missed out. And they're going to continue to miss out. I think more now with those successes and more companies starting to build into the area and so much new technology that's so diverse, right? That no one can really even be like the de facto world expert on it. I think with all of those changes, what we're seeing is, a renewed interest in kind of the technical founder CEOs. You see companies like Soleno that just announced a rather large seed round led by Khosla and the Engine. Nabiha is a young, talented CEO, straight out of her PhD at Harvard, studying physics, looking at lasers and figuring out how they could innovate in cell therapy, right? I mean, when you have a technology like that, who is even the right person to lead it? It has to be the sort of innovator themselves. So I would say that the hardest thing now that I've gotten on my soapbox about why young founder CEOs should run next generation biotech companies, I think that the hardest thing going back a few years, and I believe it's still an issue for some founders, is getting buy-in from investors to actually back people who have unproven operator records, right? You might have a great scientific record, you might have a long list of publications, you have even a pedigree from MIT or Harvard, but you don't have any experience running a research organization, you don't have any experience running a company, and so I think that it's changing, but that was definitely an early challenge for us. Many investors talked to me about who they would replace me with after investing pretty openly, and so those investors have since changed their mind. Seems like after the company has been growing and been successful, but it's I think it's still kind of there. It's probably still a challenge for some founders.
I get it. And do you think this is this going to be like, is this a problem today? Because I think 2020 was like the year of the capital investments in biotech, like a record year. Do you think it's becoming increasingly easier, or it's not that big of a difference comparing 2017 to 2021, for example?
Yeah, every year, right, has become the record year, right? That's kind of where we're at now as as an industry. Like every year since 2017 is like, no, this is the record year. And so, when that happens, I think, so that being said, yes, there's more capital. I am cautious, right? I think people need to look a little more diligently at funding numbers, right? So, okay, biotech had a record year in 2020, but what does that mean? Does that mean that Bob Nelson and Nubar Fayan and Mark Levin attracted more money to their companies? because that's what I believe it actually means, right? Like, whether it's 2 billion or 3 billion came into biotech, like, did the actual number of seed stage companies led by founders coming out of MIT, led by women CEOs, led by people of color CEOs, did that actually increase in 2020? Or did we just drive more money into the pockets of the same exact people who have kind of been sitting like a dragon on a pile of gold of the entire industry for the past 10 years? I believe it's the latter. And so has it gotten easier for, new PhDs to come out and start companies? I believe that now there's enough of a track record of technical founder, academic spin out CEOs to build and manage companies to where there is more funding available. And there are more funds. There are a lot from San Francisco, a lot from Silicon Valley, but really, just a lot of funds that are willing to come out and identify high potential people, high potential founders, and back them and mentor them and grow them as executives, right? And grow them as founders and company runners than there was even 5 years ago. I do believe that is the case, but I don't believe it's tracking at the same level of how much more money has plowed into biotech, right? More money is plowed into establishment biotech because the biotech sector is growing. I'm hoping that means that more money will plow in to, younger founders, female founders, people of color founders, like in the sector. But I think for at least the next couple of years, we're going to see, all of those numbers I take with a grain of salt in terms of like, is fundraising getting easier, or are the same people raising more money?
More money, yeah. That's a very good point. And I think this is a textbook example of lying with statistics. Right.
All right, man. Well, I think it's just like you look at the numbers and everyone wants to make the simplest, the simplest sort of correlation, right? I mean, people want to look at, more money is going in, which means more of this is happening, which means more of that is happening. But it's very rarely the case, right? There's a reason that statistics is a field of study and not a hobby, right? I mean, there are so many different ways to look at actual numbers, and it depends on what you're trying to solve for, right? Are you trying to, are you trying to make a statement that the biotech industry is booming, which it is? Then, absolutely look at the amount of money going into the field. I mean, it's incredible and it's amazing. It means more therapies will come to market. So I'm not like poo-pooing that. I'm just saying, let's back off before we think that, the entire sector and everyone equally has been able to grow and experience the same sort of new and relied access to capital.
Got it. Yeah. Makes sense. And now, if you were to give some advice to young entrepreneurs, young scientists looking to transition from academia to starting a biotech company, what would that be? What's your number one advice?
The same thing I always tell everyone is you got to network. You have to increase your network, right? And there's no better time to do it. Like every single person entering science today, like if you are entering a PhD program today, you should start networking. You should network with every single person and every, even if you're convinced, right? Many of us are when we enter a PhD convinced that we're going to try to hit that academic route. We're going to try to hit that professorship, right? Even if you're completely convinced of that's going to be a route, you need to be networking. You need to be networking. You need to be meeting people who are investors, right? You need to be meeting people who work in government. You need to be meeting as many people, especially people with PhDs that do a variety of different tasks. When we were at MIT, there was a group that was started by a collection of us that was called the MIT Biotech Group. And the point of that group was to dispel with the rumor that there are two tracks that you can take with a PhD, academia or industry. It's completely silly sort of way to think about it, right? Because you're not, academia is a wonderful route. And there's actually multiple routes within academia, right? There's adjunct professorship, and there is lectureship, and there is being a visiting professor and being able to teach after you've gone into industry. And then there's research academia, right? And there's actual kind of what people mean when they say academia. And industry, there's a million different jobs too, right? There's, there is being a bench scientist at a company. And so I think that the most important thing to do is to just meet as many people, go to as many different talks as you can, and grow your network. And don't just meet people at an event and get their business card, but like really follow up and like learn to cultivate your relationships, right? Because they're the most important thing that you'll have if you want to start a company, right? And
Right.
the classic advice is like, the best time to do this was 2 years ago, and the second best time to do it is today. So if you're a fourth or fifth year in your PhD studies and you think you want to start a company, like that doesn't mean that you haven't been networking, right? It doesn't mean that you're behind or you can't do it, right? It just means you need to start right now. You need to be going to events. You need to be trying to meet people. You need to meet people and ask them who you should meet, right? Network crawling is what I call that. And it's the most effective form to really meet high profile people, right? Meet with someone, say, this is what I want to do. This is what I'm interested in. Have a conversation. Don't just, pitch yourself. Have a conversation with someone, and then, see if they recommend, or you can even ask, like, I'm interested in XYZ. Who should I be talking to, right? Who should I be talking to in this space? And if you do that, you will quickly acquire a very powerful network, and follow up and manage those relationships. And that will be the most powerful thing to start a company, right? I mean, you want access to capital, you're going to need intros to people who have capital, right? And a warm intro is the most effective way to get people to at least listen to what you're trying to offer, right? And then you build from there. And then you have to build your team, right? You have to build executives. You have to build other managers. You have to build advisors. There's no stop to needing to know more people.
Yeah, I totally agree. And from my experience, that's been the case everywhere. Whenever the networking was lacking, success was much, much harder to get. That's even though you think you've got it all, you need help and you need other people to back you up. That's true.
Absolutely.
That's true. Well, Jacob, thank you very much. This has been super useful. Very good nuggets of information and experience. I really appreciate you coming to my show. Thank you.
Cool. This is fantastic. Thanks for having me, man. Appreciate it.
This concludes our episode. If you liked it, hit the like button, subscribe, and follow me on Clubhouse at Radu Tanasa. If you need an invite for Clubhouse, just drop me a line in the comments and I'll send you one. See you soon.